are really excited to have you all here. We will begin promptly at 12:30 Central European time, or if you're here in the UK like I am, at 11:30. So please do introduce yourselves in the chat. I believe that your mics and cameras are off automatically, but if they're not, please do make sure you are on mute if you're not speaking. We will be introducing two brilliant speakers today. So we have Maaike Gerritse, who is from the Sprint project, and she's doing her PhD with us. And we have Milla Brandão Gois, who is joining us from the University of Groningen. I find it difficult to pronounce. So we'll begin in just a minute's time. So get yourselves comfortable and we will get started promptly. Okay, so it's 12:30 Central European time, so we're going to get started. We don't want to keep any of you longer than we have to. So I'm very pleased to welcome you all to today's webinar. So as some of you know, we've been holding a series of these webinars. So this is part of the Sprint project, which is an EU-funded project. We've been running for 5 years now. We come to an end in a couple of months' time, and we've been looking at the integrated impacts of pesticides on environmental and human health. So today's webinar, we're going to focus on the impact of pesticides on human gut health, so looking at all things microbiome. So first of all, I'd like to introduce Maaike Gerritse, who is doing her PhD as part of a Sprint project, and she is based at Wageningen University Research. Once Maaike has spoken, we will then introduce Milla Brandão Gois. Milla is our external speaker for today, so we're really excited to hear some of her findings around this topic. And Milla is from University of Groningen. So please do post your questions throughout. We're going to have a nice long question and answer session at the end. We will endeavour to answer all of your questions, and if we miss any, we can follow up via email afterwards. So without further ado, I'm really pleased to hand over to Maaike. I'll stop sharing my screen so that Maaike is able to share hers.
Yes. So you should be able to see my presentation now.
Yeah, perfect. Thanks, Maaike.
Okay, great. So I'm Maaike. I'm working in Wageningen in the Netherlands, and as Charlotte said, I'm working as part of the Sprint project. And within Sprint, I've been investigating the effect of PPPs, plant protection products, or pesticides, on the gut microbiome. And today I will be presenting some preliminary results. So these results have not been published yet, but hopefully they will be in the near future. So first of all, to make sure that everyone can follow this presentation, I'll quickly explain a little bit more about the microbiome. So when we talk about the microbiome, or the human or animal microbiome, we mean all beneficial bacteria and other microorganisms that live in and on the body. So most of them are living in your gut. You have about 100 trillion microbes living in your gut. And most of these live in your large intestine. While back in the day we maybe thought that all bacteria were bad and we needed to be sterile, right now we actually know that these bacteria and these microorganisms play a beneficial role for human and animal health. We also know that there's a lot of communication going on between the gut and the brain, and also between the bacteria in the gut and the brain. We call this the gut-brain axis. And via this gut-brain axis, the bacteria in your gut actually also play a role in your mental health and in your behavior. Also important to mention is that there is a lot of interplay between the microbiome and the cells of the intestine itself. For example, if you have inflammation in your gut cells, this can again impact your microbiome, and in return, microbiome can impact the cells living in your gut. So I already briefly mentioned that the gut microbes, that your gut microbes are actually very important for your health. And they are important because they help balance your immune system and they compete with pathogens, which means that the beneficial bacteria take up all the space in your gut, and then there's no place for the pathogens to grow out and become a danger. Also, some bacteria in your gut are able to digest fibers that humans we cannot digest ourselves. And by digesting these fibers, again, the bacteria in turn provide energy for the cells in the gut. And the gut microbiome also protects against disease in a balanced state, because we know an unbalanced state is related to or linked to many disorders and diseases, including psychiatric disorders, neurodegenerative disorders, metabolic disease, heart disease, lung disease, and also skin disease. So what we mean with an unbalanced state is sometimes called dysbiosis, is that there might be less beneficial bacteria present and more pathogenic bacteria present, or the diversity might be different. And the gut microbiome can get to this unbalanced state. For example, if you take antibiotics or if you have a very unhealthy diet with not enough fibers. Or well, what we're trying to figure out in Sprint is what the role of pesticides can be to the gut microbiome. Usually if there's a small disturbance, you are able, or your microbiome is able to recover from this quite quickly. So it's okay to eat unhealthy for a day, or if you need antibiotics, you can take them. But if there's more disturbance over a longer period of time, it gets more difficult for your microbiome to also return to this balanced, healthy state. The gut is exposed to quite a number of plant protection products. This might not surprise you because there's always a very low amount of residues in your food, and there can be in water. But in Sprint we also showed that there's quite a lot of pesticides actually in house dust, and we also ingest house dust every single day. On accident, of course. We don't plan to eat dust, but it gets on your fingers, and so PPPs can also reach your gut via the house dust in your house. Of course, this is generally in very low concentrations. So the concentrations in your food and in water are quite low. There's hopefully, we think there's not an immediate toxic effect. But what we do not know a lot about is what happens over a long time span. You get these pesticides in your body almost every single day in very low amounts. And we want to know if these very low concentrations can still have an impact on the gut microbiome composition. So for the people that are not familiar with the setup in Sprint, or specifically Work Package 2 in Sprint, we included participants from 11 case study sites. These case study sites were located in Europe and in Argentina. And at each case study site, we included 6 conventional farms. So these are farms that do use pesticides, but also 6 organic farms where pesticides were not used. And at each farm, 2 farmers were included. But also in the areas of the farms, we looked for neighboring people that live near the farm but that do not work in agriculture. And also at each country where a case study site was located, we also included 24 consumers. And these were people that do not live near farms. So maybe they live in a city or in a less rural area. And in this whole process of participant inclusion, we tried to select just as many, for each group, just as many males as females. So we also tried at each farm to include 1 male and 1 female person. This meant that in total we had 72 people per case study site. So that's almost 800 participants total. And there's not a lot of microbiome data sets that are this big in relationship with pesticides. So that's what made Sprint really unique. So from these 792 people, we collected feces, blood, urine, and also they wore a wristband for, I think it was 3 weeks. And this wristband was also able to pick up pesticides from the environment and kind of measure what people were exposed to from touching with their hands. So from the feces, we were able to extract bacterial DNA. And by looking at this bacterial DNA, we are able to determine what bacteria are present in the gut. And we refer to this, what is present and in which ratios we refer to as the gut microbiome composition. That's what I'll be talking about a lot today. So that means which bacteria are present and in what ratio. Also from all these matrices, we measured what PPPs were present. We measured over 200 PPPs and metabolites, so it's quite a big panel that we looked at. And then I looked at the PPPs that were most present in each of the matrices in relationship with the gut microbiome. So after collecting all this data, there was of course a lot of data analysis to do. And the question that we asked here is, what is the link between all the PPPs that we find in these matrices and the gut microbiome composition? So if you have higher concentrations of a certain PPP in your feces, blood, urine, or measured in your wristband, what happens with the gut microbiome? Is the gut microbiome different from people that have a very low concentration? And this analysis was all corrected for country effect, because the biggest effect on the microbiome in the study was actually the country where you came from. And this is because diets are very different between all the different countries, and this has a huge effect on the microbiome. So I looked at the 60 PPPs and metabolites that were most present in wristbands, and 4 of these 60 had a significant link with your gut microbiome composition, and another 11 we consider to be of concern. And if I say of concern, it means that our study is not enough to really prove that there is a link, but there are indications, and to be sure that it does or does not have an effect on the gut microbiome, maybe it is nice to do a follow-up study, maybe an animal study, not within Sprint, but it's candidates for future research. From the 23 PPPs and metabolites that were most often present in feces, 3 had a significant link with your gut microbiome, and 4 more were of concern. From urine, we also tested 23 pesticides that were often present. 4 were significant and 1 was of concern. And from the PPPs that we most often found in blood, actually none of them had a link with the gut microbiome composition. So the PPPs that we did find to have a link with the gut microbiome composition were glyphosate, cypermethrin, DDT, chlorpyrifos, and hexachlorobenzene. These were all also described in literature. Hexachlorobenzene was not directly described, but other pesticides of this class or other compounds of this class are known to have an impact on the gut microbiome. So this was within the line of expectation. But we also identified 3 new PPPs that have not before in literature been linked with the gut microbiome composition. And these were metalaxyl, pyrimifosmethyl, and propiconazole. So from these 8 compounds, 3 were fungicides, 5 were insecticides, and 1 was an herbicide. Then I also said that we identified some PPPs that are of concern, and I found that 8 of these are also described in literature to be associated with gut microbiome composition. So with proof from literature, we can say that these do have an effect on the gut microbiome. And then there were another 5 that are also not before described in literature. So these are good candidates for future research to see if they do have an effect on the gut microbiome. So, of course, we also want to know what changes in the gut microbiome if these 8 PPPs that that had a significant link to the gut microbiome are present in higher concentrations. And we saw that for multiple of them, Prevotella was more present, and some species of Prevotella bacteria are associated with chronic inflammation. Also, we saw that Clostridium was more present for multiple of these PPPs, and some species of Clostridium are pathogenic. We saw that Catinibacterium was also more present, and Catinibacterium is also more present for multiple diseases. And one thing that was not so surprising maybe is that Intestinibacter was also more present with higher concentrations of these 8 PPPs. And Intestinibacter is also actually known to persist after microbiome disruption. So it's not surprising that if there is a small disruption, we see this one more. There were also a couple of other ones that were more present with higher concentrations of these PPPs, but these were all just normally associated with health, with a healthy state, so it's not of immediate concern that these were also more present. We also looked at which bacteria were less present with higher concentrations of these 8 PPPs, and this was a little bit of a concern because many beneficial genera, including Bifidobacterium that some of you might have heard of, were actually less present with higher concentrations of multiple of these PPPs. And a lower abundance of these beneficial bacteria is linked to intestinal conditions, metabolic disease, mental health problems, and also other diseases. Also, Bernigella was less present, and this is a group of bacteria that is known to provide resistance against pathogens. So if you have less Bernigella in your gut, you might be more prone to infection by pathogens. As I mentioned at the beginning of this presentation, there is also an interplay between the cells of the gut and the microbiome. And next to what I've been presenting out about the microbiome, I've been studying the direct impact of pesticides on gut cells in the lab. This was only for a selected group of pesticides, but we did find that glyphosate, piperonal butoxide, and cypriodinol can impact gut barrier function. And what I mean when I talk about gut barrier function is that the cells lining the gut play a very important role in keeping harmful substances, indigestible fibers, and bacteria inside your gut and out of your bloodstream. And to do this, they really interlink. They hold on to each other very, very tightly. And what happens under influence of glyphosate, piperonal butoxide, and cypriodinol is that these links that link them together are produced less, which means it might get easier for substances from inside the gut to go out of the gut, and this can lead to inflammation in the gut area. And again, inflammation in the gut area can impact the microbiome, and the microbiome might again impact the gut cells. So when we talk about the gut microbiome, it's also very important to keep in mind the environment and the interplay with gut cells. So the take-home messages for my presentation today is that PPPs can and do impact the gut microbiome. And the results in SPRINT are mostly in line with literature, but we identified some new associations with the gut microbiome, and we also identified some candidates that maybe need some more research to see if they impact the gut microbiome. The pesticides that are most commonly are used that do impact gut microbiome are glyphosate and pyrethroids, including cypermethrin. We also saw that some beneficial bacteria are actually less present with higher concentrations of certain PPPs, and some potentially pathogenic bacteria are more present with higher concentrations of certain PPPs. So yeah, that was my part of this presentation. I don't know if we're going to move on to the next presentation or if we should do some questions first.
It's fantastic, Maaike. That was really brilliant, and you made sure that we, even non-scientists could understand all of that. So thank you so much for your excellent talk. So we will be holding a longer Q&A session at the end, so after Milla's spoken, but it might be nice to just ask you one of the questions now, just to make sure that no one's left waiting desperately to hear. So a question from Milla, actually, to Maaike. So Milla has asked, did you also look at only the targeted PPP, so pesticides, found in fecal and blood samples, or also at potential byproducts of these compounds? So that's things like, I guess, additives, synergists. Did you look at that side of things as well?
Yeah, so within Sprint, we didn't look at byproducts, but we did look at metabolites. So some pesticides are broken down in your gut, or even before they reach your gut, they can be broken down inside the plants or in the environment. So metabolites can also reach your gut. So we've also looked at metabolites, but in Sprint we haven't looked at byproducts. But of course, byproducts are very important too. We did look at piperonal butoxide, which is a synergist and not an actual pesticide, but it's used together with a lot of insecticides. And as I briefly mentioned, we did see an effect of piperonal butoxide on gut cells, but not really on the gut microbiome.
Brilliant. Thanks, Maaike. So without further ado, let's hand over to Milla. So it will be really interesting to hear how your results either differ or resonate with Maaike's findings, and further evidence around how the pesticides may be affecting the gut microbiome. Thanks, Milla. Helma, just to say, yes, this webinar is being recorded, actually. So we will be sharing this via our YouTube channel and on our website afterwards, as long as both of our speakers are happy to do so. So you will be able to re-watch this at your own leisure. Okay, so handing over to Milla now. Thank you.
Yeah, and first of all, I would like to first apologize for my lack of voice. I hope you bear with me and it's not too bad and you can hear me clearly. And also I would like to thank for the opportunity to speak, and also for Maaike for the great introduction, actually, because it really pairs well with what I'm about to present. For those that missed it, my name is Milla. I am a last year PhD candidate at the group of Sasha Zernakova at the UMCG in the Netherlands. And I've done some research in the beginning, in my second year of my PhD, where I really looked into the impact of occupational pesticide exposure on the human gut microbiome, which is what I'm gonna quickly go over now with you. So I don't think I will introduce the gut microbiome, but I thought it would be nice to really look into what is there to look into when we talk about environment and the human gut microbiome. And when we look at the kind of like a world map like this that you see here on your bottom right side to the slide, you will see that there is quite a lot of difference in certain microbiome profile, the gut microbiome profiles across the world. And when you talk about different populations, of course, you go into ethnicity, you go into socio-demographic, you go into environmental factors. But when you go into within populations, of course, there's other factors that may play a role, and they're also kind of embedded in the environment, like host genetics, diet, like Maaike mentioned, some lifestyle, but also just overall urbanization, industrialization, and some other environmental factors that are even within these populations really impacting the human gut microbiome. But when we're talking about the human gut microbiome, we're also talking about an individual, and even though we're talking about cohort, we're talking about one individual. And within the individual, there's also certain things that we might take into account, like the interaction of the gut microbiome with the host metabolism, like I mentioned, and Maaike, I even asked Maaike about this, the biotransformation of such compounds, the overall microenvironment, because of course, we often talk about the gut microbiome and we often refer to it as bacteria members, but we also have to learn that we have to keep in mind that the gut microbiome really interacts with the oral microbiome or other microbes such that we often don't really talk about, like viruses or fungi or other members of the gut microenvironment. And of course, we cannot not mention the impact, again, like genetics, other diets, and other physiological systems, just like the metabolism or immune systems, all in all. And then having that in mind, why actually look at pesticides? And I know that I'm really, you probably know more about this than me. But when we, again, look at the pesticides kind of a world map, we see very similarly to the gut microbiome composition, which is quite funny. I think it's very interesting. And when we think about pesticides, we also think about its growing use in the past decades. And we often think about farms, but we have to keep in mind that pesticides are not only used in agriculture. Like Maaike also mentioned, we have low exposure to pesticides even in our house dust, but we can go further to this. All of our food is stored in a supermarket, which probably has a lot of pesticide use as well. So the majority of the population is exposed to pesticides in some level, and those are chemicals that are designed to be poisonous to microorganisms. So that makes it kind of interesting to look at, well, if they are designed to be poisons and we are ingesting them, what do they do to our gut microbiome, right? Well, considering pesticide exposure, we have to look into, like Maaike was looking at, unintended exposures or airborne. So if you live close by, often the wind will take some of these. If you spray the pesticides, the sprays, and if it's windy like it is in the Netherlands, then airborne exposure to these pesticides is also a factor that should be accounted for. The unintended, like the house dust or the diet. But also we have to look into occupational exposure, which is actually from the farmers or from the people that are handling these chemicals. These are often repetitive, though with a bit of varying levels of exposures. And then particularly at that, I was really thinking that this occupational pesticide exposure might be a very nice thing to study on the effect of the gut microbiome, especially because of its profile being repetitive, being a chronic thing. May help us with some of the associations and to untangle these links. So these studies really aimed into looking whether occupational pesticide exposure is associated with certain changes in the gut microbiome. For this, I used, we used 7,000, a little bit over 7,000 participants from the Dutch Microbiome Project, which is part of the Lifelines, which is a big, large populational cohort from the northern of the Netherlands. We did metagenomic sequencing on the fecal samples of these participants, and for those, I've already skipping all the QCs here because I know this is not a microbiome specialists. So we're skipping all the metagenomic sequencing. This is a very deep sequencing technique, and we selected based on this 168 bacterial species. I selected this also based on the prevalence of these species to avoid having certain individual biases. So I kind of selected based on the presence of this bacterial species in at least 10 or 50% of the exposed group. And then I looked at the same species also on the unexposed group. And then I retrieved the occupational exposure to pesticides as well. So occupational exposure to pesticides was a bit of a tricky thing to do. The first thing we do is we classify the jobs based on tasks and responsibility, and this is done so by the ISCO, which is the International Standard of Classification of Occupation. Based on this, there is also this developed method, which is called the exposure matrices. Among different exposure matrices, which assign probabilistic levels of exposure to different chemicals, there is the Aloha Plus job exposure matrices, which then goes into exposure to pesticides, heavy metals, and some other heavy fumes. But it assigns levels of exposure such as no, low, or high exposure to each of those job classification codes. Using this ISCO code in the Aloha, then we were able to go back to the Dutch microbiome participants and look into the employment questionnaires, the ISCO codes and the gut microbiome, and retrieve this occupational pesticide exposure matrix, where we have the data on pesticide exposure to pesticides, herbicides, and fungicides and insecticides, both in a factorial and also in the long term. So I also looked at how long are the participants employed under the same job, what is the classification of exposure of that particular job, and what type of tasks do they have, and then probabilistic, how would they be exposed to these chemicals. Here also importantly, we're not looking at one particular compound. We're not looking at one exposure compound to one outcome. Here we're looking at the overall class of pesticides, herbicides, and fungicides and insecticides, because we also understand that usually those compounds are often used together. There are often used one or two pesticides, and then they change in the next year or so, but then they keep within the same class of pesticides, herbicides, or fungicides. Then the first thing we do is, what is the exposure profile? We have within our cohort around 300, we have 309 participants that are exposed to at least one type of pesticides. Most jobs or occupation comes from indeed farmers and some wood-related work, but actually there's a third group that was really, really present, which is material handling and transport. This is freight workers or workers that are working on just the overall storage facilities of supermarkets and stores overall. Majoritarily, these exposures often follow this chronic long-term exposure, which really hints towards a very low but steady perturbation to the microbiome. When we look into the gut microbiome and the association to these pesticide exposures, the first thing we look into is the overall microbiome composition and the overall microbial diversity, which is the amount of microbes that you can find in the samples. Those are significantly decreased after exposure, whether this being low or high, but there's not a lot of difference between low and high. So there is a decrease in diversity when you compare to no exposed people to exposed people, but the levels of exposure are not often very significant. Most of this is the statistical significance really did happen on pesticides, fungicides, and insecticides. And interestingly, herbicides were actually not significant, although you do see a small little trend. This is probably likely due to numbers, to lower statistical power, because herbicides are not as used as pesticides and insecticides and in fungicides when we did our analysis. Though all four types of pesticides that we have measured, they were highly correlated to each other. So they're often also co-occurrent with each other. The overall, when we look beyond the overall diversity, so diversity is usually linked to how many microbes there are there. When we look into the beta diversity, and beta diversity here is the overall profile, whether you have, for example, a group of microbes that are more prevalent, and this is the majority of your microbes, and you don't have a lot of low abundant, or whether you have a lot of low abundant and not a lot of highly abundant microbes. So the overall composition beyond just who is there also follows a similar trend of associations, but because of the low numbers, the plots are very not informative, so that I kept just the diversity. So then we went into the impact of particular microbial species, and then we see, interestingly, two types of changes of certain bacterial species after pesticide exposure. One is where we look into the proportion of presence, and here you see the figure A and B. This is basically telling you that certain bacterial species are more present in people in which they were exposed to certain pesticides. So in both cases, this is true in irrespective of the type of pesticides. in comparison to the non-exposed group, where here is in gray, where you see that the proportion of presence is very low in comparison to the other groups. The other types of profile of changes in the bacterial composition, and here it's particular microbial species, happens when we look into, for example, the figure C and D, which here is the abundance. So how much of that particular microbe is present in the gut of those participants. And here we see that certain abundances actually decrease upon pesticide exposure. Also, again, in all levels, sometimes, like the case of herbicides, it's not very significant after adjustment for multiple testing, but you still see a certain significant, you still see a trend towards significance. So when the microbe is very abundant, those are often very abundant bacteria that we see differences in the abundances. So those are very abundant bacteria like Bacteroides doré. This is a very well known, very well described bacteria, and you see that there is a decrease of the abundance of that bacteria after pesticide exposure. These are often, so Bacteroides doré, for example, the decrease of its abundance, it's very well associated to other health outcomes or gut disruption, so the dysbiotic, disequilibrium of the gut. And you do see the increase of low abundant bacteria in the presence of them because they're very low abundant. So usually the overall abundance, it's very not informative, but looking at presence, absence of these very low abundance, then you see that they increase. This usually pinpoints towards a sort of a environment where the gut microbiome is already disturbed, and then other low abundant bacteria have kind of a space to grow. So this is sort of how this microdynamics can happen within the gut upon pesticide exposure. So just to summarize a little bit, what I've showed you is that occupational pesticide exposure in the Netherlands is often a chronic low-level exposure. We do see that there is pesticide exposure that are associated with a decrease of gut microbiome diversity and the overall microbial composition of participants that are exposed to pesticides. We do see that there are decreased abundance of both Bacteroides doré and Alistopis feingoldii, which is previously linked in other studies to other health outcomes. We do see also that low abundant microbes, which are not very well described in literature, are more present, but their occurrence may be linked to the impact of these toxicants in the gut environment. And then I just like to thank you, and I'm open for any questions and for the discussion.
Brilliant, fantastic, Milla. Thank you so much. And it's great to see that your findings have lots of similarity to Maaike's as well, in that pesticide exposure does indeed affect certain microbes in terms of both diversity and abundance. So we'll move on to our Q&A now. So if we could have Maaike and Milla and perhaps Vera as well present, just to make sure we're able to answer all of your great questions. So I'll get started. First of all, with a question to Maaike, and this was from Milla again, actually. So Milla asked, did you look at whether there's a difference in the combination of pesticides found? Some PPPs were found together, and were they linked to a stronger impact in the gut microbiome?
Yeah, so this was very difficult to assess because, well, we're looking at 4 different matrices, and it's really quite individual what combination you find. So each individual has a different combination, therefore it's very difficult to make groups to compare with each other. I can say that, pyrethroids are known to have an impact on the gut microbiome, and we do see that if you have more pyrethroids, if we combine all the pyrethroids that you have in your feces, the effect does become stronger. So that's one way to look at it, just to combine everything from one specific group. And yeah, it's within expectations that the association becomes stronger. And that's also what we saw for perpetuates. Yeah.
Brilliant, perfect. Milla, did that answer your question? Just to make sure. Yeah, it sounded like it did. So that's brilliant. Thank you. So we'll move on to a question from Bartos. So Bartos asked, well, there's a bit of context first, so I'll explain. So Bartos said, EFSA identified data gaps regarding the human gut microbiome in its risk assessment of glyphosate, and yet the substance was ultimately reapproved in the EU for a further 10 years. So from your perspective, should these gaps, or the fact that the EU regulatory framework doesn't currently require detailed consideration of microbiome impacts, should this be a cause for concern? How significant are these gaps in terms of our understanding of potential health risks? Is the current regulatory toxicology keeping pace with emerging science on the microbiome? So a few related questions there, but essentially, yeah, we're asking, should EFSA be considering the human gut microbiome more when they're thinking about risk assessment?
Yeah, so this is quite a difficult discussion because the microbiome is so diverse and complex that it's really difficult for us to say, oh, this person has a healthy microbiome and this person has a disturbed microbiome. It's really quite personal, and what's healthy for one person can be unhealthy for another person. This means that with where we are right now in microbiome research, it's also really difficult to directly implement this in toxicology assessments, because even if we see something happening, we cannot immediately assume that it also has an impact on health. However, knowing how important the gut microbiome is to human health, and that it's actually linked to so many different health outcomes, I do think we all need to talk about the impact of environmental chemicals, including pesticides, on the microbiome. Definitely.
Excellent. Thank you, Maaike. Milla, do you have anything to add there?
Yeah, I would like just to add that it's important to keep in mind that in gut microbiome studies, the directionality of the effect is still open for discussion and is still not very well agreed within the field. So we don't know, and we do not have a lot of information without in vitro studies to look to know whether the impact of health of microbiome on health, is it causal or is it consequence? What is the directionality of this? And the same holds true when we're looking at pesticide exposure. Is it the cause? Is it the consequence? What is the directionality of this? So it's important to keep that in mind, because when we talk about microbiome, people often want to already apply it, but the causality is still up to debate.
Yeah, absolutely. Thank you. That's perfect. So Pascal says, Obrigado, Milla. Did you include residential information in your statistical analysis? And do you know if calibration measurements were made to assess occupational exposure?
Yes, so indeed, I looked at residential information based on the urbanicity level and as a proxy for urbanization, and how would that is it closer to a farm? Are those workers close to a farm or not? And I correlate that with pesticide exposures levels, and those are not very well correlated. This also is because pesticide exposure, we have this line of thought that it happens only in farms, and that is not true. That is not what I see in the data. So we do see a lot of pesticide exposure coming from cities, coming from industrialized people, and the residential and the occupational addresses are often not the same. So I did check residential exposures, but it didn't come out as a huge thing. In regards to the calibration methods, the job exposure matrix is a sort of validated matrix to measure these or to assess a probability of exposure. So there has been some papers out when each of these job exposure matrices come out. They are very peer reviewed and validated with actual bio tracking of samples. I did not do bio tracking of these samples because this was a particular retroactive study, so I did not have information on the actual urine samples or actually seeing and measuring these pesticides of these participants particularly.
Great, brilliant. Thank you, Mia. I mean, this just indicates the need for further research as well. I think us as scientists, we always have more work to do, and each of the projects we do does have a limit in terms of its scope. So yeah, that's brilliant. So Helma, I think this is a question for Milla again. So Helma has said pyrimidine methyl is present in almost all bread and other cereal products in the Netherlands. Did you find a large gradient of this in the feces in your concentrations at all?
I actually think this was from Maaike, because I do not have fecal concentration.
Yeah, so I just had I saw the question pop up. I had a quick look at the data. Actually, we don't really find pyrimidine methyl in feces. This could be because it might be metabolized somewhere in the body, and then we don't actually see this compound in feces. Where we do see it is in wristbands. So that means that via air or via touching things, the wristband picks it up. So yeah, I agree that there is quite some exposure. It might be for apart from bread and other cereal products, but in fishes we don't really see it that much.
Brilliant. So Milla and Maaike, do you have any further questions for each other? It's quite a nice opportunity because you're carrying out quite similar research. So I just wanted to give you the opportunity to ask each other some questions.
Oh, that's a good one.
If you have any, there's no pressure. I know Milla's already asked quite a few of Maaike, so.
Yeah, not right now. Actually, it was quite clear.
No, definitely. Yeah, no, I think it's been a couple of really great presentations today, so thank you both. So I think we can begin to draw to a close unless we have any last minute questions. So just to make you all aware that we are hoping to hold a further Sprint Ask the Author webinar within the next month or so. We're hoping that this one will be about our indoor dust results. So our indoor dust findings have received quite a bit of attention recently. So we think that this will be a really exciting webinar. Just this weekend, in fact, The Guardian published a piece about this research, so they've picked up on it. So please do sign up when we share the registration form. It's best to either sign up to our email list via the Sprint website or stay tuned via our various social media channels. I'll post a few links in the chat shortly. Another date to save in your diaries is our Sprint Final Conference. So as I've mentioned, Sprint is coming to an end. So we're holding our final conference in Brussels at Leidtre Coterie on the 25th of June. So please save the date if you'd like to join us. This will be an in-person event with an option for streaming remotely. So please do consider joining us. And in the meantime, we'll be sharing this webinar via our YouTube channel and website. So please do stay updated and informed. And yeah, without further ado, I'll allow you to leave and go and have some lunch. And thank you all very much. Thank you. Bye.
I think there's some extra questions that came in. I think we can still answer them.
Oh, we can do it. Yeah, we're finishing early if we don't. So I think that's brilliant. Excellent. I see these questions. So Pascal has asked, do we know if there are any follow-up studies in our research group on evaluating the impact of pesticides on health, considering microbiome changes as a mediating factor? So any sort of research coming up?
So in my group, we're actually focused on host microbe interactions. Usually we look a bit more at causality, actually. And I know one of my colleagues might use some of the pesticides that I have left over in his study of Parkinson's. But as far as I know, in my research group, there's no big follow-up study in the planning. But yeah.
Perfect. Milla?
Yeah, I think there are quite a lot of growing market for in understanding the environmental exposures. Actually, I know of a few consortium that are like I'm involved in the Exposome consortium, which looks into environmental exposures, not only pesticides, and how it impacts health, both in adults and in infants as well. The problem is that the impact of health outcomes, it's quite a noisy thing to study because the impact of health actually varies per person. So it's very noisy and very tricky to actually derive what is disease status and what is health. Usually we associate health on the lack of disease, but the disease in and of itself have a lot of variation within the population. And pesticides, and again, you're talking about a very varying type of data, both from the pesticides, both from the microbiome, and actually also from the health outcomes. There are a lot of initiatives on studying this. Currently, I can name Exposome NL. There is the Initialize project. There are some also cohorts both in Finland and in Sweden. You can there are quite a lot. There has been a lot of biomonitoring also in the UK done with the UK Biobank. So there's quite a lot that has been done. But again, when it comes to linking pesticides and health in and of itself can be quite tricky. Some of them might have cardiovascular consequences, some of them might have neurological consequences, and it's often co-occurring with other comorbidities such as smoking, alcohol abuse, and some other things. So to clean out this, it's quite tricky. Researchers are doing that, but it's been a ride.
Yeah, brilliant. Thank you. So yeah, lots more research to do and lots of research happening by the sounds of it. So that's great. I think this also quite nicely answers Anna's question. She's mentioned specifically the causality side of things. So do you have any other further initiatives to share around how we link changes in the gut microbiome to diseases? So causality side.
So one thing that I know has also been done a lot in my group is actually looking at metabolites that are produced or can be produced by microbes in the gut, and then looking at the effect of these metabolites on health. So this is all in the lab, so on cells or on organoids. But it really requires to go very detailed and look at one specific aspect. It's still very difficult to talk about the microbiome as a whole and health. Yeah.
That's brilliant. Perfect. Fantastic. So I think we have now answered all of the questions. So that's fantastic. And yeah, Pascal says thank you to you both for your nice answers and presentations. So yeah, brilliant webinar. Thank you both so much. And yeah, to our listeners, please do stay in touch. Drop us an email with any further questions and stay tuned via our various channels for our next upcoming webinar and our final conference, which is really exciting. Amazing. Thank you, everybody. Bye.